Okay, DNA engineering, sky's the limit. You can do anything you want. And then without it, we wouldn't be able to analyze the function of all of these proteins encoded in the genome, because there'd be so little of it. So we can use this then to express these rare proteins on the milligram scale if need be, like structural biologists do. You can go both ways then, so you can go from DNA to protein or protein to DNA. And now here comes this reporter construct again. So thrown in there just to drive home the fact that you can use these to see when and where a gene is expressed. And in addition to that, you can use it to dissect the effects of these individual, one, to identify them, and then two, to dissect the effects. Okay, so that's what's shown here is LAC-Z, but you can also do this with GFP, and so then you can identify specific cells in a live animal. So that's a reporter construct as well. So you're not trying to dissect the promoter, but you're just trying to see where is this gene expressed, and you can use it as a marker to follow these cells through development. Okay, mutations best reveal the function of a gene, so you can do that through site-directed mutagenesis, produce that mutant protein. Okay, model organisms, model mice, not trivial, but very powerful. So you can ask, what's the requirement of a certain gene through knocking it out. You can also replace it with specific mutations. Say you want to determine what's the role of a specific residue, amino acid residue, in a protein. So you can make that mutant and test its effect through gene replacement. Or you can throw in gene addition, a transgene, like the GFP. Not easy to make. You can't just transform in like you can bacteria. So you have to get these embryonic stem cells, and then using these micropipettes, get these embryos. It's extremely laborious. Then you've got to breed the mice till you can eventually get one in the germline, hopefully. Then this helicase mutant mouse just shows you how dramatic these effects can be to test for the role. And then this final slide then, RNAi. I was talking to Abiel about this. I mean, you could have a whole lecture on RNA interference. I mean, this is just a huge concept, and we're just talking about one aspect of it here. But this is a principal one where you throw in these small inhibitory RNAs, and they lead then to this degradation of a template or a transcript. So it silences. So it's gene silencing, and so you're inactivating. So it's like a knockout, but it's just on the molecular level. Okay, so board questions. Yeah, see, these are just super easy. Site-specific endonucleases, separate strands, southern blot, everybody knows that. Ligase, everybody knows that. Polymerase, chain reaction, useful for what? Amplifying a specific DNA sequence. Well, duh. But we saw several examples of it, didn't we? And then DNA complementary to RNA. Okay, reverse transcriptase. I think we all knew that too. But that's single-stranded. How do we get to double-stranded? DNA polymerase. Okay, so
you said that they're not going to be the same. You said they're not going to be the same. You're going to switch them up. You're going to switch these up a little bit. Did you say?
Yeah.
So you're going to. Was that what you just said? Is that what you just said? Are you going to add a little bit more to it or something?
Yeah, I'll just.